So then I need for PowerPoint.
Good morning and warm welcome to all the participants joining from all over India and abroad as well in this international webinar on stem cells regenerative medicine organized by the Department of Biochemistry. Let us start this webinar with induction by Dr. C.R. Utra, Dean of Research.
Good morning, viewers, and a pleasant warm welcome. ES Educational Charities was established in 1983 by the esteemed industrialist 3 Swami Kanna, and 12 institutions has blossomed under the ES group banner. Theivanai Ammal College for Women was founded in 1989 and accredited by NAC with A grade in the third cycle. The college adopts CBCS system with focus on academic excellence, vocational skill development, value orientation, and sensitization to community development. Signing of quality regional, national, and international MOUs add globalization to the education on the campus. The college has been recognized by national and global bodies. DST has sanctioned funds for conducting Inspire program for school students for 3 consecutive years and later on by FIST for establishment of central instrumentation facility. The college has a number of academic enrichment programs and good track record in extracurricular activities. The college emerged as 84th college in India among self-financing colleges ranked by Education World. The college is powered by modern technology and is automated for its day-to-day functions. At present, there are 3,000 students on roll and the college runs 12 UG programs, 8 PG and 5 PhD programs. On behalf of the college, I welcome you all for this international webinar. Thank you.
Thank you, madam. Now it's time to welcome you all by myself, Sridevi, head of the Biochemistry Department. First and foremost, I would like to welcome the renowned resource person, Dr. Akon Higuchi, Chair Professor of National Central University, Department of Chemical and Materials Engineering, Taiwan. Welcome, sir. I feel honored in welcoming our visionary and proactive Secretary, sir, of ES Education Charities, Mr. S. Sendil Kumar, a technophilic person, though is not here. Welcome, sir. It's my pleasure in welcoming our dynamic registrar, Dr. E. Soundarajan, sir, who always encourage us in all our endeavors. Welcome, sir. Next, I would like to welcome our effectual principal, Dr. M. Brinda, for this webinar. Welcome, madam. I also welcome Dean of Research, Dr. C.R. Putra, Vice Principal, heads of the department, faculties, and students of my institution. I am delighted in welcoming all the participants from various reputed universities, institutions, research sectors, and industries. Once again, I welcome you all. Coming to the introduction of the chief guest, I am glad in presenting the brief biography of the resource person. Professor Higuchi started his career as assistant professor in Meiji University, Tokyo, and joined as a guest researcher in Riken Advanced Science Institute, National Research Institute of Child Health and Development, and a visiting professor in King South University. He received his PhD in Tokyo Institute of Technology in 1985. He was a professor in Seki University from 1993 to 2007. He received Sophie Memorial Award from Society of Fiber Science Japan in 1994, Seki Academy Award from Seki Alumni Association in 2003, and Nanotechnology Outstanding Contribution Award by National Science Council Taiwan in 2013. He is interested in the development of materials for stem cell research. He established purification method of hemopoietic stem cells and mesenchymal stem cells from umbilical cord and adipose tissue, respectively by filtration method through polymeric porous membranes. He is also developing biomaterials for stem cell culture. He published 3 articles in chemical reviews as first and corresponding author with a high impact factor The Nature or Science, and 2 articles in progress in polymer science. His professional activity of scientific society includes editorial board member of World Journal of Stem Cells, China, counselor of Chemical Society of Japan, Division of Polymer Science, editorial board member of Journal of Membrane Separation Technology, Canada, associate editor of Physics and Chemistry of Stem Cells, German, editor-in-chief of American Journal of Tissue Engineering and Stem Cell, USA, advisory board of journal materials in Royal Society of Chemistry, the Asia Pacific Council of Tissue Engineering and Regenerative Medicine International Society. Editorial board member of Journal Stem Cells Research, Development and Therapy USA, and editorial board member of Journal and Cytology and Tissue Biology USA. He organized and chaired sessions in international conferences such as International Chemical Congress of Pacific Basin Societies Pacific Chem 2015 USA, Termis AP 2013 Annual Conference Shanghai and China, the 12th Asian Bioceramics Symposium, National Cheng Kung University, Taiwan, International Symposium of Materials on Regenerative Medicine, Taiwan, the 7th Conference of Asian Membrane Society, Korea, International Congress on Membranes and Membrane Process in Holland, the 2nd Taiwan-Japan Symposium of Nanomedicine, the 2010 International Chemical Congress of Pacific Basin Society, USA, Asia Conference for Biomaterials and Stem Cell Techniques, and 5th Conference of Asian Membrane Society, Japan. With more than 30 years of experience, he has published more than 300 journal papers in reputed journals, 13 books, 50 patents, attended 100 invited lecture at international and domestic meetings, 3 funding reviewers, presented 621 papers at international conference, and 20 invited papers in Japan and Taiwan. We are glad and honored to have such an eminent speaker in today's webinar. We welcome you, sir, once again. Now I invite Madam Principal, Dr. M. Brinda, for keynote address.
Very good morning and warm welcome to all. First of all, I would like to extend my sincere gratitude as Secretary Mr. S. Chandilkumar for providing permission to have this webinar. It's my pleasure to welcome our Registrar Dr. E. Soundararajan. It's my pleasure to extend a cheerful welcome to our resource person, Dr. Akon Higuchi, Great Scientist, Chairperson, Professor of National Central University, Taiwan, Fellow Royal Society of Chemistry, England, Fellow AIMBE, USA, is in the Advisory Board, Journal of Material Chemistry B, Royal Society of Chemistry, editor of Open Physics. Here, I would like to specify, a few points about Dr. Akon. Actually, in 2016, actually, we had a plan to have an international conference in my previous college. That actually it was a collaborative program with the Thiruvallur University. When we are discussing about the resource person, with the former VC, Dr. Murugan, actually he suggested Dr. Akon for the webinar, for the conference. So he gave his number as well as the email ID to me. When I rang, immediately Dr. Akon accepted our invitation and came to India, and he has given a very good presentation in our international conference. Actually, nearly 3 days he was with our department teachers, and he enjoyed all our Indian foods. Moreover, if you say he is a very simple, very punctual, kind-hearted, well-disciplined, and loving and lovable person. Now in our college, when we are discussing about this webinar with biochemistry HOD, actually I suggested the department HOD about Dr. Akon. He was very happily accepted, and when I sent a mail, immediately Dr. Akon responded, and now he's with us now here. Thank you for inviting our invitation, sir. Next, we'll see the keynote address. Stem cell therapy is known as regenerative medicine. It promotes the repair of response of disease, dysfunctional or injured tissue using stem cells or their derivatives. It's a process of replacing or degenerating human cells, tissues, or organs to restore or establish normal function. It enables scientists to grow tissues and organs in the laboratory and safely implant them when the body is unable to heal itself. Regenerative medicine has the potential to heal or replace tissues and organs damaged by age, disease, as well as to normalize congenital defects. I welcome you all to this webinar to present and discuss the topic concepts and the questions regarding the scientific approach to the regenerative medicine. Thank you so much.
Thank you, madam. Next, I request our Registrar of hESC Educational Charities, Dr. E. Soundarajan sir, to give felicitation.
Most respected resource person, Prof.Dr. Akon, Chair Professor of National Central University, Department of Chemical and Material Engineering, Taiwan, respected Principal, Dean of Research, HOD Biochemistry, faculties, participants, good morning to all of you. First, let me thank the organizing committee for this opportunity given to me. Our college secretary, Mr. S. Kumar sir, conveyed his best wishes for every success of this webinar. Once again, on behalf of management, I extend a warm welcome to our resource person, a wonderful personality, Professor Dr. Akon. In spite of his tight schedule, kindly accepted to act as resource person. Thank you and a hearty welcome to you, sir. I feel proud to say that Biochemistry Department got approval from Thiruvallur University to introduce PhD program from this academic year 2020-2021 onwards. I like to congratulate the HOD and the faculties for taken effort to bring this program. I like to appreciate the organizing committee for choosing a right topic in this webinar, stem cells, regenerative medicine, which is the need of the hour. Researchers and doctors hope stem cells studies can help to understand how disease occurs, generate healthy cells to replace diseased cells, that is regenerative medicines, test new drugs for safety and effectiveness. Researchers have discovered several sources of stem cells such as embryonic stem cells, adult stem cells, and so on. I'm sure that this webinar is eye-opener to the participants, and I request all of you to listen till the end of the session to gain more knowledge on stem cells and regenerative medicine. Wish you all success. Thank you. Thank you all.
Thank you, sir. It's my privilege to welcome Dr. Akon Higuchi, resource person of the webinar, for honoring address. So now the dais is yours.
Okay. Okay. Hi, hello, everyone. Thank you very much for your kind introductions. Okay, so then I'd like to start for today's talk. Okay, so can you hear me? Can you see me? Okay, should be okay. Okay, so this is actually for kindly for already for for introduced. So just briefly, I'd like to for explain my background. I graduated from Tokyo Institute of Technologies for 1979, and I fortunately I received several for awards, like for fellow of the AIMB or Royal Society of Chemistries, and mainly actually for in our lab, actually for we are much focusing on to publish in high impact journals. For we published for 3 chemical reviews, that is for impact factor 53, and also progress polymer science, that is for 22.6, and also progress material science, that is 31, and a lot for biomaterials, that is for impact factor for 10s. And actually I was for organized for several for international meetings, and also for organizer of the session organizer, and also chairman. So this is for for its kind of for so one for introduction of my papers, that is for over 330 for citation, and still for this is for highly cited papers for Web of Science, and actually for front cover pages for my pictures for this one. So then, now actually I'm a chair professor in National Central Universities, and just briefly I'd like to introduce my universities. And my university was located in the northwest of Taiwan, and capital is for Taipei. So that is for short distance to capital is 40 kilometers, just only less than 1 hour drive. And also it's very close to the international airport. That is for just only less than 30 minutes for driving. And here we can find our students. Undergraduate is for around 6,000, and the master course is 4,500, and the doctor course is 1,500. So actually for our university is research-oriented university, and especially for engineering, and it's very strong. So then, okay, so I'd like to talk about for today's story for human embryonic stem cells and induced pluripotent stem cells and their clinical trials at first. So first, think for briefly, I'd like to introduce what is for embryonic stem cells and also what is like for induced pluripotent stem cells. So embryonic stem cells, that is for typically for sperm and oocyte. Then it's going to fertilize the eggs. Then it's going to differentiate. Then finally it generate like us for humans. But for before it's going to be for humans. So then divided. Then there is a for cavity inside of the blastocyte. Then we will get for inner cell mass. Then if we culture for special for dishes, so for material scientists, to development this kind of cell culture dishes to guide the stem cells and to keep the pluripotency and also differentiation, it's also for, very good for topics. And only for some special conditions, we can get the pluripotent stem cell that is called for embryonic stem cells. Of course, for embryonic stem cells can differentiate any kind of our body tissue cells. So that means we can use for clinical therapies. To use it for this. For ES cells. And also for 2007, one genius guy that is Professor Yamanaka actually developed for the stem cells that is very similar to ES cells. That is for induced pluripotent stem cell. Because for our body cells was already differentiated, but he actually inject just only 3 or 4 pluripotent genes, typically OCT4, SOX2, KL4, and CMYC. Then it's amazingly actually for at least for 10 passage, if we cultivate on the cells, then that becomes the stem cells that we call for induced pluripotent stem cells or iPS cells. But these iPS cells is very similar characteristics to the ES cells. So then again, so this ES cell, embryonic stem cells, and iPS cells can differentiate to any kind of our tissue cells, such as for hematopoietic stem cells that can be used for blood disease, and also for cardiac cells that is for heart disease, and also for hepatocyte that is for liver disease, and pancreatic cells also can be differentiated. So that is we can use for diabetes patients. So this for ES and iPS cells is for a lot of for possibility to differentiate. But another thing is the difficulty is for this kind of for variety of differentiations. So that means we should be guide this stem cell for specific differentiations. So that is not only for actually for development of the small molecules such as growth factors and some inhibitors, but also cell culture biomaterials can guide the stem cell for differentiations. So just this is for briefly, we can see some history of ES and iPS. That one typically for together we said for pluripotent stem cells or PSC. So actually for the history is not so long. For example, ESL was, human ESL was developed just 1998. That is for Professor Thompson. And again, Professor Yamanaka developed for human iPSL. That is 2007. And actually for he received for Nobel Prize for 2012. So that is just only 5 years he got the Nobel Prize. So that means we can find for iPS is a very great impact to our society. And if we consider for some clinical trials, 2010, one is for very famous for U.S. company for Geron, was try to spinal cord injury, and also another for persons was for used for ESL derived regenerative epithelium RPE for the treatment of eye disease, that is for AMD, that's age related macular degeneration or Stargardt disease. That one later I'd like to explain for these issues. And also for we can also doing this kind of AMD patient for treatment using iPS, but they are using for cell seed of the iPS. And also for cardiac for projector cell was for actually embedded to the fibrin patch. Then for one company, it was for trying to cure for myocardial infarction disease. And also another case is a diabetes patient. So actually for ES and IPS, for treatment, for clinical treatment, it's not so much. Actually for just only for disease. But if we consider for another stem cell, we call for MSC, that is mesenchymal stem cells. These mesenchymal stem cells are such as bone marrow stem cells and adipose-derived stem cells, and also for teeth, for teeth for stem cells. So that is much more for differentiated cells. That one was for actually for a lot of cases for clinical trials. So one reason is this kind of MSC, mesenchymal stem cells, are limited to the differentiations, typically for osteoblast, chondrocyte, and for myocyte and adipocyte. So this is for mesenchymal, mesodermal lineages. But this is also a good point. So differentiation ability is for limited, so that means for much more easy to control the stem cells. So then it's a very rare case to be tumors, for cancer cells. So this is why they are for huge amount of clinical trials. But another issue, so this kind of bone marrow stem cells were mainly using for clinical trials, but not so much for effect. But so one most, predominant, and everyone is going to accept for the, for some effect of MSC for treatment. That is for GVHD. This is graft versus host disease. So that is for immunosuppression and can be for immunoreaction can be suppressed by using for MSC. So especially if our body was injured, there is for inflammation reaction was generating. But if we inject to the MSC, this kind of inflammation reaction can be suppressed. One reason is here for right side. Here we can find natural killer cells and B cells and dendritic cells, and proliferation and differentiation can be suppressed. Another thing for activated T cells is going to apoptosis. That is that activated T cell is going to dead. So then MSC was working for the immunosuppression. So this is very powerful for MSC for effect. So then again, in our lab, we are for treating for mesenchymal stem cells and ES and iPS cells. Here we can find for ES and iPS based products, in clinical trials. Actually, for not so different. Actually, for this is for in summarized for 2015, but actually for 2020 it's not so different. Then here we can find for what kind of purpose for this disease. This is here we can find for dry or Stuttgart. Actually, for Stuttgart and AMD is very similar. So this is all IDGs. So here we can find IDGs, IDGs from the top. So most 2, 3, 6, 8. I think for 9, for this product, that is all relating for IDGs. Then next is for severe heart failures. For that is for patch of the cardiomyocyte progenitor cells. And also next was for diabetes. So that is for insulin secreting for cells. And the last two, that is osteodendrocyte for injections. So actually, so the point is there are a lot of for treatment for ITGs. So that reasons also I would like to talk for later. Before that, so clinical trial for spinal cord injury using for hESCs, I'd like to explain for this one. And actually for our, for spinal cord, there is for, there is a several disc and there is for numbers, like for neck, that is cervical, and also for back, thoracic, and much more lower part is lumbar. So then, actually for this bone, not only these bones, we know inside there is for nerve fibers. This is central nervous systems. So typically, if we have accident, this spinal cord is going to damaged. Then some part of our body does not move. So then, actually, for it's very severe. If we find our body do not for move, so then we need some treatment. But for here we can find, for if we have some accident, here we can find the x-axis for the time. Actually, it's shown by date. Then first action for 1 week is going to acute phase. And the acute phase, if we actually for inject the like this kind of stem cell or stem cell differential cells, it's not so good ideas. Typically, but if we inject chronic stage like after for half year or 1 year or 2 years, in this case it's too late. So then typically for this injection is around less than 1 week, like for 5 days after the injury, for 5 days like to 8 days, like this one. Again, for after for acute inflammation stage was for decreasing, then we can inject this kind of stem cell for product. So then here we can find actually for maybe for number 3 is needles, and this is a part of the spinal cord. So we should be very carefully to inject these needles. Then we are going to inject oligodendrocyte. Oligodendrocyte was for differential to the human ES cells. This is for product was for originally developed by a Geron company, but now it's going to shifting this technology to another company. Then it's called this is AST OPC1, that Asterias Biotherapeutics. That is for now they are for proceeding for clinical trials. So another example is for heart disease. So myocardial infarctions. So in this case, actually for this is for paracrine effect. That is for ESL-derived cardiac progenitor cells. This cell was for entrapped with a fibrin patch. So then this fibrin patch was transplanted to the patient. So then from this fibrin patch, this for ESC derived cardiac progenitor cell was secreting several growth factors. Then this, because of these growth factors, this for myocardial infarction was going to be cured. So then this is again for right figure shows for fibrin patch. And left side here we can find after for 2 months. So then black, for bars here we can find for this treated by this fibrin patch. Then ejection fraction. Ejection, that means for heart blood, how can be eject. So then that is for typical case is around maybe 55%. So then compared to nothing for treatment, compared to control, few percent, more than for 5% can be, for improved by using this kind of patch. Again, in this case, this fibrin patch, this is for cardiac progenitor cells derived for ES cells. That one was going to, secrete for growth factor. Actually, for growth factor is actually for it's kind of for medicines, and the growth factor is a protein medicines that can be secreting for constantly. So this is why this patient can be cured by using this kind of stem cell therapies. So another treatment of diabetes. So diabetes, actually for differentiation, we call for beta cells. So the beta cell differentiations, that is actually for in our lab. Also, we tried in past. This is a little bit for difficult for differentiation. ESL to differentiation to the several for tissues like a cardiomyocyte or cardiac progenitor cells, that are not so difficult actually. Actually, for in our lab, we are for routinely differentiate. And ESL or iPS to differentiation of MSC, mesenchymal stem cells. That also not so difficult. And the good point of ESC derived for MSCs, that one for passage is for over 20. But typically, if we isolate for MSCs, typically for 8 passage, so that is for 2 months, only 2 months can be survived the cells. However, if we derive for ES or iPS for MSCs, so that is more than 5 months, we can use it. So that is for great point. And also, if we isolate the patient for MSCs, it takes times. But if we for stock ESC derived MSCs, that is on the shelf, that is for any time, to the patient, we can use. So that is for great point. Not using the patient original cells, but ES derived cells or iPS derived cells. And for then back to these stories, for this diabetes for treatment, we need for beta cells. Beta cell was the secret of insulins. But the point is, even if we differentiate the insulin secreted cells, it's not enough. The cells should, respond to glucose concentration. So for us, we eat, for food, like for rice. Then, the glucose concentration in the blood was enhancing. Then our bodies, for naturally, insulin was going to secreting and for suppress the glucose concentrations, in the blood. So the point is, we should actually for differentiate for this kind of beta cells. That means, for wise for they have characteristic of the sensor. That means so depend on the glucose concentration. Then glucose concentrations high, the insulin was secreting much high. But the glucose concentration is low, then insulin secretion is for decreasing. This kind of cell to differentiate, that is for very difficult. And then if we done this kind of I mean, if we finish to make it for this kind of good for beta cell differentiate from ESL or iPSL, then like this for companies, this should be entrapped to this kind of polymers. This is because if we inject the ESC derived for beta cells, but this one was, for actually for specific for type of the tissue, we call for HLA, human leukocyte antigens. If this does not match, so immediately, our, for immune system is going to attacking and, actually for kill this kind of cells. So protect this kind of immune cells, we need this kind of protection. So then we need this kind of porous materials to cover it for this one. So then next step, I'd like to talk about for treatment for AMD, age-related macular degenerations. So then let's see for especially for developing countries. So old people, many old people has this kind of disease. So then, so left side, especially for the, for center of the, eye visions, that is going to, cannot be seen because for photoreceptor cells was damaged. Actually, for my father was also this disease for this one, much more severe one. So then, let's see. Actually, for finally, we'd like to, for introduce how we are going to treat for this kind of MD by using for ES or IPS derived for cells. And there is for dry AMD and wet AMD. But actually, for we are mainly focusing on the dry AMD. And so we will see this kind of structure of eyes. For light is coming, let's see for light fields, light is coming first, ganglioid cells and bipolar cells, and then it will for simple name is photoreceptor cells. And then for last part, here we can find RP, regenerative epithelium. Actually, for light can be sensed by cone and rod. Then this information was transferred to the bipolar cells and ganglion cells. But RP, regenerative epithelium, is very important because RP is one is for repair, actually for repair the core and rod cells, and actually for our strategies, very strong light like a UV light, core and rod was damaged. Then actually for this kind of damaged cells was removed by RP, we call for phagocyte. Then actually for new, for cell of core and rod is going to regenerating. So if RP, regenerative epithelium was damaged, we cannot make clean, in our eyes. So it's going to fall damaged much more. So typically, if we inject the regenerative epithelium, then vision loss was going to, treated. So actually, for another reason is why ES and IPS cell for treatment was much focusing on this kind of disease. Because for eyes, actually for eyes, isolated place. So then immunoprivileged natures. So that means for just one cell line of ES cells or IPS cell can differentiate RPE. That one can be used to any kind of for patient. Of course, in this case, we need for to patient should take some immunosuppressive medicines. But if patient for have immunosuppressive medicines, he or she can see for much more improved for visions. So this is why just only one cell type is for possible to using for clinical for treatments. So this is why a lot of for company was focusing on this treatment of the disease using for stem cells. So then here we can find again for regenerative epithelium RP layer that maintains photoreceptors health. So this is why typically we are going to inject RPE for treatment of AMD patient. And then again, ESL or iPS RP can rescue photoreceptors and prevent of vision loss. Again, photoreceptor cells is a cone and rod. So this is for one report, it's and also very famous one. For Professor Schwartz was reporting for using for ESL derived RPE. So then first actually for ESL was for differentiate RPE. And actually for in our lab, also we are now challenging to RP differentiations. And this is also, relatively for difficult one for this one. And then here we can find for left top. Here we can find for black spot. That is for regenerative epithelium. And another thing is a good point is for one cell culture dishes such as a six well that is a 6 holes. That one actually for we can get 1.5 to 10 to 8 regenerative epithelium. So that one actually we can use for 15 patients. So that means for it's not really necessary to produce for huge amount of RPE. So it's very nice for treatment. And RP has a for right top. Here we can find we call for cobblestone structures. And finally, we are going to check RP. Typically, actually, we are using for RP65 for staining, but in this case, bestotrophins or PAC6. PAC6 is for neural cell marker for this one. So then they are going to for check. They succeeded to, differentiate. So then this is for, before for clinical trial, they are doing the, for animal experiments to inject to the mouse. Then here we can find for right hand. So this is fluorescence. This is for, for marked for human for proteins. Then here we can find human RPE. It can be for inject to the mice that we can find for this one. So then this is for for human trials, and I think for this is black spot. For small for squares, then this is for larging. Then here we can find this black spot because RP regenerative epithelium has for actually for for color, it's kind of black colors. So this is why to injections for cell can be found from the black colors. Then after treatment, here we can find they are going to check. For actually for 300 until 360 days. That is for 1 year. Then here we can find after for transplant. For 19 days, so almost 3 months. Then here we can find the, this visual, acuity is going to enhancing. So more than for 10. So that is for here we can find for improvement because of the injection of the RPE, which was derived for ESL, can be found. And for this one, it's okay. For next one, so this one was using for iPS previous study was ESL differentiate for RPE, but for in Japan, because Japan was developed for iPS, so then iPS derived RPE was for transplant to the humans. And in this case, they are using for cell seat. So please show for M. It's a very small, but for it's kind of for squares, very small squares. So this for few pieces of the cell is going to inject to the humans. But unfortunately, actually if we actually for previous for Schwartz, Professor Schwartz case was the injection of the suspension cells. So no actually for another supporting material such as it's not they are not injections the gels, just only cell suspension. So one is for very easy, but for these cells to for remaining actually for their report is high almost for 92%, but typically for transplanted cell was remaining less than 1%. But for IKS, it might be high, but we are not really sure. So then for cell seat, the cell cannot be shifting because this is for the, it's kind of for solid. So then it's much more efficient to remaining to the eyes. But unfortunately for results, not so actually for improvement. So currently from the literatures, actually for cell suspension injections, it's much more powerful compared to cell seat for technologies. This one. So then this is for overview. In fact, I talked this overview of ES and IPS. Then I'd like to now it's for almost 50. Just briefly, I'd like to explain actually for our lab what we are for thinking and what we are doing. Actually for ES IPS, that is mainly focusing on the treatment of eyes because eyes, as I already told, immunoprivilege. So then if we consider for ES, IPS to, for transplant other tissues for the top one, so we need to prepare for many cell lines. Even if we consider for very special for 100 cell lines, then probably for covering to the 80 or 90% of the patient in that countries. And also depending on the country, this kind of tissue type is different. And the second one was for iPS for IDS. That is for using for patient cells we make for iPS, then it's going to differentiate. So that one was original idea for 2007 to maybe 2010. But however, now we recognized it take for a long time. I think probably for it to take for 1 year. And also, the point is for not only for culture and differentiations, we need for verify no contamination of the virus, and also for it's safe to get for permissions. It's very for difficult for to pass for regulations. But if we have just only one cell lines, it's kind of for dream cell lines. We call it for universal for IPS or universal ESLs. Just only one cell line can use to anyone. So that is a fact actually for in our lab was for challenging. So in this case, we should be considered for immune system in our bodies. So we have actually for most of us cells we have this kind of HLA typing, human leukocyte antigen systems, and this one actually for we have for example class 1 that is A B C E F G, but mainly immunosuppression immunorejection can be found for in HA class 1 of A, B, and C. And here we can find the gene types. That is for A is 6,000, B is 7,200, and C is 5,800. And these numbers actually for year by year it's enhancing. So that is we do not know really numbers. because so many people, so it's difficult to check all of the people. And this even the HLA class 1 is for even A, we will get the genes from mother and father. So even for one for array was for the same, the other was for different, then it's going to immunorejection was generating. And also we should typically for organ transplantations, they are checking A, B, C, and DR. So then actually for typical case for for organ transplantation, for for this is for A, B, C, DR. So that is for mother and the fathers. Then we get for 8 types. So then if it's for matching for like a 5 or 6, then it's going to accept it. But for this kind of patient, should take for immunosuppressive medicine forever. So then one idea is, okay, why not knock out? That is for disappear. Knock out this class 1 and class 2. But if we knock out class 1 and class 2, then these cells actually for it's kind of for no phase. So then natural killer cells, who are you? We cannot find no one. So then, it might be not humans. So then natural killer cells is going to attacking. So, completely knock out class 1, class 2 is not a good idea. So there are several ways, and nowadays, for gene editing system is pretty nice. This is CRISPR-Cas9. And the typical case is, for, last for, right side for bottoms, class A, class B, is knockout, and just only one type of class C is going to remaining. So then matching possibility is enhancing. So actually for there are several for similar types. So if we just remaining for one array of HLA-C, so typically if we prepare for 9 or 10 very special for cell lines, then this is matching most of the peoples. But another question, for them, what happened for babies? For babies and mothers. So finally, they have for immunorejections. But for during the baby, baby does not, show for immunorejections, because baby needs for nutrition and oxygen from mothers. So in this case, for baby cells, actually for expressing some specific genes like a PDL1 and HLA-G and CD47, and especially for CD47, these proteins don't eat me signals. So then one method is for overexpression of PDL1, HLA-G, and CD47. So this kind of background we consider for for baby cells is some key point to generate for universal iPS without genetic for modifications. And then we checked for HLA class 1 and class 2. And most of the cell was expressing, of course, for HLA class 1. And HLA class 2, some cell was not expressing, so we are much more focusing HLA class 1. So then, because for we mentioned about for baby cells, even actually for amniotic fluid stem cells express HL class 1, but do not show immuno for rejection, because for this is baby cells to for accept for mother cells. So then we are focusing to using amniotic fluid. And also actually for we use for amniotic fluid for mixing two different for donors. So then actually for this one, in this case, if we mix amniotic fluid from for two different for mother cells, then actually for strong immunorejection, that was for generating actually for survival cell does not show strong for immunorejection. This is for our hypothesis, and actually for we success for this method. So then we get umbilical fluid for several donors and mixing and to cultivation. Then we can get umbilical fluid stem cells. So then we just briefly to showing, and also normal method just only for one donor. For this is a single type. We also make for umbilical fluid stem cell. For morphologies, no difference. Then we are making for iPS. So in this case, again, in this case, for for this kind of mixed cells to to transfect the pluri-potent genes, we can make iPS. Then iPS cell can characteristics this kind of colony formations. So then here we can find to keep going to for cultivation. Then we can get this kind of colonies. This is single cells and mixed cells. Single cell is much easy to make for colony, so that is make for iPS. But finally, we can get for iPS. So we need some specific technique. Using for if we use for mixed for umbilical fluid stem cells to using. So that is for actually for this is for universal iPS cell for this one. So then we just check. This is really for iPS. That is just like a ESN. So pluripotent protein expressions such as Octo4, SOX2, and NANOC and SSEF4 is expressing, and also possible to differentiate 3-germ layer. So the definition of this kind of iPS and ESL is for differentiation ability into the cells derived for 3-germ layer, such as mesoderm, ectoderm, and endoderm. Ectoderm is a neural cells, and endoderm is for actually this is liver cells, and mesoderm this is for muscle cells for this one. And also we did the experiment for teratoma, so that the in vivo can also differentiate for 3-germ layer cells. Then not only for iPS, this iPS was going to differentiate to the cardiomyocyte. Then here we can find beating cells can be fine. I'm not sure for whether it's moving or not. Sorry. I think if we take time, we can find this should be moving, but this time it's a little difficult to see. Can you find? Okay. So anyway, this should be beating. Then to checking for cardiomyocyte marker was expressions. Then this is for main event. Our universal cells do not show HL class 1, class 2. So that means these cells, even the differentiate, actually for differentiate cells also do not expression. But for actually for here we can find single cells or in ESIPS expression, but our universal do not express. So then to more checking, not only for this kind of HL class 1 to not expressions, not only for flow cytometries, actually for we are using for another person's for blood. Actually for this is my blood to taking, and we are getting for mononuclear cells. Then we are going to treating. Then here we can find normal ESL, normal iPS cells. Here we can after treatment. So here we can find the red cells. This red cell is a dead cells. But here we can, if we treat, our universals do not show any dead cells. So then here we can find the verify for universal cells. And also not only for iPS or this kind of for ESLs, here we can find for cardiomyocyte differentiate to the normal ESL or normal iPS cells. And also our for universal iPS differentiate cardiomyocyte, and after treating the mononuclear cells, iPS direct cardiomyocyte is going to dead, but no dead cell, even allogenic for mononuclear cell was treating. So then here we can be fine for we verified that our cardiomyocyte I think for immunorejection, privilege And here we can, after mononuclear cell treatment, here we can find CD4 beating for our universal cells. So then I'm almost results. Actually, for amniotic fluid contains for few universal amniotic fluid stem cells, which can be reprogrammed into universal iPS. And we propose one method to, to generate the universal iPS without genetic modification. That is for mixing for FSC and actually for attacking each other. Then remaining cells was for for immunoprivilege. So this is always happened for in human societies. Not say for brave guy is remaining. So weak, but avoids for fighting. That kind of guy was survived for a long time in development. This is the same thing. So not fighting for for rejections. That kind of cell can be survived. And for this universal IPS do not show HL class 1, class 2, and also no immunogenic reaction by allogenic for different persons or mononuclear cells. And this is for my laboratories, for our lab members. And this is for our laboratories. And sometimes we will get for internship from USA and other countries. So we are enjoying to, for lab work. And the students make this kind of birthday card. Okay, thank you very much for your attention.
Thank you, sir, for enlightening us the basic concepts of stem cells, growth of stem cell technology, benefits of mesenchymal stem cells for regenerative medicine, and ongoing research works, especially for myocardial infarction, Dept mellitus, macular regeneration, and retinal degeneration. Really, the session is highly informative and interesting. We are very much delighted to have you in this webinar, sir.
Thank you.
Principal Madam, would you like to share anything?
Professor, I have a question. How does the MSc influence immunosuppression?
Yes, sorry.
Yeah, how does the MSc, influence immunosuppression?
Influence to the immunosuppressions.
Yes.
So if the cell has immunosuppression of the MSC.
Yes.
Okay, so that one was for example, if we have organ transplantations,
Okay.
then typically we have for immunorejections. But this one was for typically we are using immunosuppressive medicines, but this is also very severe side effect. So then if we also inject for MSC, then we can reduce the immunosuppressive medicine, lower concentration. So then that is we can avoid for side effect. And some person
Yes.
Yes.
Yeah, now I understood. Yes. Thank you so much. Thank you, Professor.
Thank you, Madam. Before going to the conclusion, I request all the participants to fill the feedback form, which will be shared in your mail. And immediately after filling the feedback, you will be receiving your e-certificate. Okay. To propose vote of thanks, I call Dr. J. Kalimadi, Associate Professor of Biochemistry.
Good afternoon to all. All good things come to an end in life. So is the international webinar on behalf of TACW. I take this opportunity to propose the vote of thanks to those who have directly and indirectly continued this webinar on stem cell regenerative medicine organized by TACW Biochemistry Department. At the outset, I thank our chief guest and resource person, Dr. Akon Higuchi. You are really enlightened the scope of different areas of stem cells regenerative medicines. We are thankful to our Honorable Chairman, Secretary for their motivation and his words of encouragement. His able guidance has always encouraged us. We are grateful to our Registrar. He is a constant inspiration for all the activities of TACW. And I would like to thank our principal for her enthusiastic support of the TACW. A special thanks to our Vice Principal, Dean R, Controller of Examination, Dean A, Dean S, HOD of Biochemistry, teaching and non-teaching staffs for their unflinching support on coordinations. Okay, a heartfelt thanks to our students, research scholars, professors for active participation. With these warm words and a kind message, we move to the end of today's international webinar. Once again, I say thank you one and all. Thank you. Thank you, sir.
Thank you, sir.
Thank you, sir. Professor, thank you.
Okay, thank you. Bye-bye.
Okay, bye-bye. Bye-bye.
Bye-bye.